I'm going to spend some time talking about at least assemblies we have today and where we're going, which is making these pan-genomes happen and peeling these things apart into diploid genomes, as Seth's been saying, and then how we can apply these to other medicinal organisms that are clearly on many people's radar. There we go. The slide point is working. So we're going to be talking about both Cannabis Sativa and Psilocybe Cubensis today, and some of the sequencing efforts we put forward to these. A bit of an outline here is we're looking at these pan-genome approaches as well. We are also going to be mapping more of these Illumina short sequencing reads to these pan-genomes, because you can get a lot done with these things even with short reads once you have good references. Of course, everyone prefers to have the longer reads, but if you're short on dollars, the Illumina stuff can get you many more for the same dollar. And you can, in a lot of the genes that are well mapped and understood, you can make sense of out of these shorter reads. And then we're also looking at ways to further reduce the cost from whole genome sequencing down to just targeted regions. If you follow the history of human genomics, we went through this period of time where we were sequencing exomes. And that was because it could bring the cost down to sequencing just the genes of interest if you wanted to survey particular regions of the genome for even lower costs. And then if you bring it one step further, as Seth had shown as well, we've built SNP chips that people can use to survey 85,000 markers in the cannabis genome so that you can deploy those at even further numbers of plants at a reduced cost. And the ultimate reduction of this approach of surveying less and less is to then make PCR markers that can survey a single or 5 loci per assay point. So once we have, I'm going to go over some of these strain-seq panels that we've recently developed and recently put public so that anybody can order these panels and use them at their university if they need be. So this becomes a community design panel that people can use. And then we're also going to look at some pathogen response characteristics in the cannabis plant, and then move over to other fungi that are very much interested in this community, such as Psilocybe cubensis. Okay, so before the pandemic, which seems like yesterday, right? We put out this Jamaican line reference that Seth was speaking about. This was 1,000 times more contiguous than the short read assemblies that existed before it. But we had to use a technology from PacBio that isn't quite what they have today. It's known as CLR technology, which was a continuous long read technology that could do about 80 kb reads. However, the noise rate in the reads alone, they're probably around 90 to 95% accurate. So there was a little bit more error inside of those reads, which can be complicate things when you're dealing with a very polymorphic plant. So as a result, we really hammered this genome with 125x coverage. You don't need to do this anymore. The HiFi technologies they have, you can do this in 30x coverage, and we'll touch on one of these HiFi genomes we've done as well. And the N50s come out around 3.5 megabases. And so out of reference to the reference that's currently in NCBI that Seth had hinted at being somewhat shuffled, we've also seen that same problem. The N50 numbers for that are around 750 kb, and they piece together those contigs with Oxford nanopore, which doesn't do a very good job in repetitive regions. It gets very, very long reads, but it has very high error rate, and so you can get really confused in polymorphic plants piecing together the chromosome numbers. The chromosome numbers in NCBI have frequently changed as a result of us trying to organize this picture as a community over time. And some of the new references that are coming out are helping to clarify this. We'll touch on one of them today. So this is a great paper from Herb Bogan, who went through all of the references that were available at the time of this publication, and you'll see some of them that are really old were done only on Illumina and 454. Some of those we put out in 2011, and then they get better over time as we graduate to these longer and longer read sequencers. But what you're looking at here is a really important quality score that Seth had touched on, which is called a Bosco Score. You scan these genomes for genes that you anticipate every particular plant should have. We just have a catalog of these. We know that all plants should have these number of housekeeping genes, and if you're missing some of those housekeeping genes, you probably don't have a complete reference genome. And of course, these databases of Bosco genes are different for every type of genus you attack. So when you go into fungi, you got to change your Bosco database. But what you're looking for is to have the least amount of green on this slide, which are missing genes. This is the part of the genome that you failed to sequence because you couldn't assemble it correctly, or it's missing for other biological reasons. The region that you see in gray is actually, what that's showing you is that the sequencers are starting to phase the genome. That means that they're splitting apart the maternal and the paternal haplogroup into their own unique contigs because the mother and the father genome were different enough from a sequencing standpoint that the assemblers turn those into their own unique contigs. And then the blue area are all the regions that have been what Seth called a synthetic haploid state. The mother and the father genome were so similar, we just called one haplotypic state, and we didn't need to split them apart. Okay, so this is moving toward phasing these genomes, and it's very, very important in cannabis, more so than it is in human, actually. Okay, so what you can see from those slides is that the later the technology you used, the more complete the reference is. It has nothing to do with the cultivar that each of us chose or how smart we are. It just happens to be with how late we decided to start sequencing these things and what part of Moore's Law that we were on. So we did run something through HiFi, which is what Seth talked about many years ago, and this gave us N50s, which is the average size of the contig. It inflated it out to over 5 megabases, and the Bosco scores are getting up at 97%. So that's about as good as you can, as close to finished as you can get without having to do a 20-year Human Genome Project finishing episode. You'll notice the Human Genome Project recently closed and finished everything, and they did that really because of these HiFi sequencing tools. They threw some Hi-C and some BioNano in there as well, but I think if you ask those researchers what was the #1 thing that really changed and closed the human genome, they would tell you it's the HiFi sequencing chemistry that really made that happen. So you can take these sequences we have and align them to CS10, which is a type 3 plant. Jamaican line's a type 2 plant, and you can see there's not 100% agreement. There's some crossover here between all of the different homologies, and those differences at the time, we didn't really understand were those true biological differences between the plants or were these assembly artifacts. I think we're beginning to appreciate that they may in fact be assembly artifacts as more and more other people publish these same observations. The other thing that you can see with HiFi is you can see the length of telomeres now. Now this is something that was really cool because I have never seen 40 kb telomere sequences before in my life, and I've been sequencing since 1996. But out pop these telomeres with really not a whole lot of effort. You can see they do vary based on chromosome. They're quite different in length. Some are a couple kb, some are 40 kb. I bet this is a bit limited. There could be some in there that are 60 kb, but we didn't really have the read length and HiFi to be able to resolve them at that point. But this is something to think about with cannabis sensitence and to think about with cloning age. If you hyperclone things over time, the telomeres likely shrink like they do in mammalian genomes, and this may give you a sense of like a molecular clock on the plant. Okay, so we did some work with Oregon CBD and urifins as well, where we ended up doing a lot of genotyping across an F1 population in Jamaican lion, and that helped us build these linkage maps. So you can estimate, the actual genetic distance between two different markers in a genome is a proxy for how close they'll recombine. But really, the devil's in the biology. You have to actually go and do the crosses, and then you see how frequently these things actually recombine in biology, because the genes at the end of the chromosomes have a tendency to cross over more frequently than the ones in the centromeres. And so these linkage maps are important to calibrate how likely is it that a gene will be co-inherited with another one. You can't just use a megabase scale on this. You have to kind of convert it to centimorgans, and this project helped us do that. And these categories and buckets that you see in here in color are the concordance that we have with the Hi-C assembled version of this genome. This is similar to the dovetail work that Seth was demonstrating. This is a chromatin capture technique that buckets chromosomes into the proper place when you start with a collection of hundreds to thousands of contigs. So we've got a nice understanding of that thanks to some of that work. We have a few folks in Massachusetts that have gone and tissue cultured this. So yes, Jamaican lion can be tissue cultured. There's nothing weird about it. We've built SNP arrays off of this reference, and we've also built some of these mini exome tools. And I look forward to, if better references come out in the next year that are perfectly phased across 100 genomes, it'll be a boon to the industry, and we'll readily adopt them, because it's a real challenging problem to work on references that are dated in any way. Okay, so we have taken then an approach here of taking Illumina shotgun sequencing, because it's more affordable, to blast through another 40 genomes and map it to these references. And this is in a preprint right now form that's up in BioArchive. And what you can gauge from here is the type of variation that you see amongst all these genomes. All right, this is more variation than we see in human genomes, and human genomes are much bigger. So there's about a variance every probably 50 bases between the hemp lines, and maybe every 90 bases between the type 1 lines when you compare them to the Jamaican Jamaican lion reference. And if you get into the Y chromosome, this thing jumps to like a variant every 25 bases. The Y chromosome is much more polymorphic, and it's the largest chromosome out there. There's flowering genes on there. There's a host of things in the Y chromosome that we have to pay attention to. The indels are the insertions and deletions, and just beware the other axes up there that you're looking at is the indel. Oh, missed that. Well, we're not going to go back. I'll tell you about indels later. So the two different axes there, one is SNPs and one is indels, and there's 10 to 15 million variants from a single nucleotide level, and there's millions of them from an insertion and deletion standpoint. And when you get into structural variations, the numbers go down from there substantially. But there's still a large portion of the genome that is in structural variation. The most obvious thing in cannabis genetics right now is what's known as the BTBD allele that Etienne de Meyer originally described, but didn't know the genetic location of. That's been fully resolved now, and I'd probably argue that maybe 90, other than some of the exotic stuff that Seth has in his library, most of the cannabis genetics out there, the THC and CBD ratios are governed by this. We're going to touch on some of the rare things that they found as well that can be an exception to this rule, but basically the presence of or absence of CBD or THC synthase dictates the chemotype of the plant, with the exception that he mentioned from Zerpel, which is that if you clone these genes into yeast, they make a portfolio of cannabinoids even though they're supposed to be dedicated to making one. So CBD synthase makes like a 21 or 25 to 1 ratio and can leak some THC synthase. So we have to be careful about that. We also have to be aware of the fact that there are many other cannabinoid synthase genes in the genome, and we don't know how much those leak yet. But we're presuming they probably leak, and they probably aren't specific to making a single cannabinoid. They probably make predominantly one cannabinoid by orders of magnitude, but they probably leak other ones as well. So this heat map here is a list of the, did I get that right? No, I think that's backwards, that's forward. Well, I don't know how to make the laser pointer go on this, but what you're seeing on the right-hand axis there are the cannabinoid synthase genes, and you're seeing the presence of absence of those in red and blue. So the Jamaican lion references are over on the right, and you get this kind of light blue color because they all have those genes. What you're seeing in the red and blue are the disappearance of certain genes or the inclusion of them. And what Seth had mentioned, what we had found a few years ago, is there is a very large deletion that covers the cannabichromine synthase genes. There's many of these genes in the Jamaican lion reference, but when the deletion occurs, it takes out some neighboring genes with it. And these neighboring genes are known to be associated with pathogen response. Now, one approach to breeding perhaps low THC plants, a really clever way is bringing in the varins. Another way is maybe breed plants that have less of these other cannabinoid genes so that you don't risk the leakage from these synthase genes leaking other cannabinoids, potentially THC, into your process. So some people are breeding to get rid of the cannabichromine cassette, and that it's possible this can back you into a corner in that it takes out some pathogen response genes. Now, down the lower area in yellow on this map, you can see that there's kind of a scattering down there. That means that's the part of the deletion that isn't consistent plant to plant, and some have variable lengths in this deletion, which means some deletions of CBC synthase can occur that don't knock out these genes. So you gotta be careful when you go about trying to knock these things out. You can also extend this to pathogen response genes on the right. There's another list of chitinases, thaumatin-like proteins, and MLO loci that are in here. These are things that are published in hops to have a big role. Well, we also had done some iso-seq sequencing in this. That's RNA sequencing with PacBio. What that had demonstrated is the most highly expressed gene in the genome were chitinases. These are things that dissolve fungal coats and fight off pathogens. What we noticed in some work from Colorado Seeds is they had a copy number amplification of some of these pathogen response genes. One was a chitinase and one was a thaumatin-like protein. So we took those genes, we cloned them, and we started running beta-glucanase assays to look for how effective were these genes after we expressed them in E. coli. Did they still work, and would they actually attack glucan? And so we ran some assays that showed that yes, the cannabis cloned enzymes do in fact attack these glucans. And then we started putting them down on petri dishes on these tabs of paper to see if it would retard or change the growth of various fungi that are known to infect cannabis. So you can see a penicillium species there, a fusarium species there, and we can in fact put these enzymes down and they slow the growth of these pathogens that are known to affect cannabis. Now, this sounds like a great tool to spray on plants, but the hydrogen peroxide is pretty cheap. Enzymes like this, even though they're cloned in E. coli, are very expensive. We decided there may be a better tool for this that fit into our pipeline is, can we use these in our pathogen detection tools to actually do a better job of cracking open yeast and mold so that our tests don't miss any of those things. So here's an experiment we did where we just put these enzymes after they were cloned out of the Jamaican lion genome into E. coli, expressed those enzymes, put them into broths, and then spiked various different amounts of fungi in there to see if these enzymes would actually liberate more of the DNA or whether they would kill some of these organisms. And we can in fact see a massive shift in the quantitative PCR results when we have these enzymes present. That tells us that we're actually doing a better job at retarding the growth of these particular fungi. This was done actually with aspergillus, and we're getting a 4 to 64-fold reduction in the amount of aspergillus that grows in a given time period when these enzymes are present. So these have now made their way into our kits. They've been in the field for a while now. Our new PathoSeq products actually have this enzymatic lysis involved, or at least a derivative of this enzyme involved, that helps crack open these yeasts so we get a better job at actually surveying the pathogens when we're trying to make safety tests for the pathogen field. Yvonne's going to speak about this a little bit more tomorrow. But these kits have now made their way through AOAC, at least the aspergillus, 4 different aspergillus species are through, Salmonella, Stech. We're just in the process of moving total yeast and mold and total aerobic count through. But there's a lot of complexity to actually doing this correctly, which she's going to touch on tomorrow. But yes, there are genes in the cannabis plant that have basically given us some wisdom on how to better survey the fungi that might in fact infect it, which is quite cool. Okay, so what can we do from the sequencing here? If we're going to build a panel to focus on sequencing certain genes, which are the ones that probably matter the most to the community? So we put in the cannabinoid synthase genes, we put in the terpene synthase genes, we put in the flavonoid genes, the chitinase, thaumatin-like proteins, the MLO genes, a lot of the other genes in the genome that have been annotated as being pathogen resistance on Jamaican lion. And then we also put in some flowering genes from both the X and the Y, lots of markers across the Y, and some random ones distributed across the genome, so that the SNP chips and that the sequencing tools that try to target these things for lower costs are actually targeting some genes of really, really high interest. This is now a community design panel. Anyone can order it and use it at their local university. You don't have to necessarily sequence with us. You can just get the kit yourself. But you can use the software as well. So this is a public software service on Cannapedia. You can import your samples here if you'd like, and it will analyze it according to the this thing's even higher than 1605 as of today, but these are all the genomes that are currently in there. It will give you an estimate of how rare this genome is compared to everything that's in the database. It will also walk you through how much X to Y sequencing we see. This might be pertinent when we start looking at hermaphroditic traits. Right now, there isn't a real clear signal as to what genes drive this, so we've decided that we're just going to monitor how much of the reads come out of these systems that match the X or the Y, and maybe some patterns will emerge out of understanding this. And then we also go over those genes in the BTBD allele that Etienne Demeyer described, is that we want to look at the coverage of these genes, and the presence of absence of these genes gives us a really good predictive model as to whether you're going to be a type 1, 2, 3, 4, or 5 plant. We can gauge that all from the sequencing alone, with some minor exceptions that we're going to touch on here. This is something that we frequently had customers come to us saying, we sequenced with you and it's a CBG plant and you called it a type 1 plant, what the hell's wrong? And we dig into it, invariably we would find this variant. So by the time this happened like the 8th or 9th time to us, I decided to write a white paper about it to explain to everyone why this variant matters. And when I put it up on the portal, I noticed from Seth, maybe a week later, that they had already published about this a month earlier in the pandemic, and I missed the paper. But they also find that this variant is in fact attenuating THC synthase. And it's amazing because it's a single variant that seems to be driving this. We've happened to find this variant is always in concordance, or I should say in linkage with another variant known as P333R, which itself doesn't seem to have any dysfunctional activity on the enzyme. But this is a location in the genome that Zerpel had tagged as being really critical to the glycosylation of this THC synthase enzyme. And so it's very likely that the damaging is because it's adding a glycosylation site to the THC synthase, and that's probably making it a dysfunctional version of it. So you can have a copy of THC synthase in rare cases that some of these type 4 plants have, and it's completely ablated by having one of these polymorphisms. So simple copy number analysis may miss this, and you need to get some SNPs in there. These are the 8 different strains that kind of haunted us about this, that we had sequenced over time that have this same variant. And I think one of the more interesting ones is Doug's variants, because Doug's variant was actually a THCV line, so it did make some THC, and it made it as THCV, but a little bit more in THC. So what is it doing? Well, it turned out it's actually heterozygous for this variant. It's got one copy of THC synthase that works. It's got one that's broken. And if you look at the chemotype of this, it also has half the amount of the cannabinoid is actually CBG. So there's a bottleneck in synthesis here. When one of the genes is knocked out, we can't convert all the CBG into THC, so we get a buildup of CBG, and we get a little bit of THC and THCV as well. Very interesting. So as I mentioned before, the platform will give you some heredity information in terms of what else these things are related to, if it's common, if it's uncommon, if it's rare. The SNP chips are now integrated as well, so if you run SNP chips with Eurofin, we can port that data in, and we can still call the copy number variants off of SNP chips because there's enough variants in these genes that we just hypertile those genes and watch how many of the assays drop out, and it can give us an information about whether it's CBD dominant, THC dominant, and either whether it's male or female as well. So there's a bunch of different tools you can use here. The best is obviously whole genome sequencing. The absolute pinnacle is what you're seeing Seth do, is doing HiFi sequencing and building a reference. It's more expensive and more time intensive, and you need very good high molecular weight DNA. You don't always get that out of a hole punch from a plant. However, you can get a variety of these things from like a hole punch of a plant. A very small amount of DNA can be used for these short read sequencers. There's varying degrees of resolution here. There's whole genome shotgun with Illumina. There's doing this 18 megabase panel with Illumina. It's 10 to 18 depending on how deep you go. And there's an 85K SNP chip that you can use. Varying degrees of information that you can pull off of this, and they all integrate well with this Cannapedia tool. So what else can you do with this? Well, once you have a really good reference, you know what your pathogens are. And that's very important when you're looking for viruses, because oftentimes viruses just look like retroviral elements in genomes. So you want to have a really good reference so that things that don't map to that reference, you can then go categorize and say, okay, what do these things look like? This was actually done really elegantly by the group at Phylos and Darkheart. They went and actually did RNA sequencing, and they were able to assemble those things and find new viruses. And sometimes it's very helpful to first map all your reads against the references to pull out the things that are related to the plant, so you can play with the things that are clearly not from the plant. So we've gone and built lots of virus tools. There's a list of them up there. We have released these 5 ones so far, and the other ones are all made. And the reason we haven't released them is we've not found molecular evidence for them yet in the plant, other than tobacco streak virus that was just published out of Colorado. However, it published and it was only 83% identical to what was found on tobacco. So one of the lessons here is that even though these things share names from other plants, oftentimes when you find the virus in cannabis, it's so significantly diverged, it probably deserves a new name. That's some of the problems we have. Is in releasing these assays, we don't want to release them until we actually see the DNA sequence evidence of the virus that's on the plant, so we know that our primers actually hit them. We have all these working on positive controls. We have not found evidence of them in cannabis when you look at the ones that are on the pipeline side. The ones on the left, we actually have some evidence of them being in cannabis, and I'll show you some examples of this working really well. So we work with a few grows across the country. Some that were suffering from hop latent viroid, some that were suffering from lettuce chlorosis virus, and one that was actually suffering from both. And they were quickly able to scan their mother plants, identify the plants that actually had these viruses and viroids, cull them, and move and get their grows back to a particular place of productivity. This worked very well and very quickly for them. However, we did learn a couple lessons in these things. Here's some other pictures of LCV that was making a real mess of some of these grows. One lesson you learned is that the phenotype isn't always evidence, right? When you have one of these diseases, it doesn't manifest in like a clean, like that's an obvious HLV and that's an obvious LCV. These things sometimes have pleiotropic conditions, and they can each disease can look like one another. So the visual confirmation doesn't necessarily tell you what it is. The other thing that we noticed in this chart here, what you're looking at in blue are hop latent viroid assays that were run across 12 different mother plants, heirloom mother plants from Colin Palmer that were like 30 years old. He did not want to get rid of these things. But one trend you'll see is that when the plants are actually infected with hop latent viroid, they're screaming hot with CTs in the 20s. I would not advise people to be culling really important mother plants with very late CTs, because those could be noise from PCR, they could be contamination, or they could be something very interesting biologically, that when you have something that's 100,000-fold lower viroid content, you might be stumbling across a plant that actually has some form of resistance that you want to like put aside and maybe study a little bit. What we don't want to have happen in the industry is aggressive use of PCR and people start weeding out all their mother plants and suddenly the genetic diversity is just imploded. So the CT values are really important right now for us to monitor these and understand what we're looking at from a viroid load level. The other thing to do is to sample more than once. Okay, when we did this with Colin, since these plants were some of the original like early ChemDog and Sour-D lines, is we said, all right, we're going to PCR this thing in triplicate. We're going to do it every 3 days, and we're going to do that for 3 weeks. So we're confident that we know this mother plant is toast, and there's nothing we can do other than maybe go to a tissue culture lab and sort it out. If you're dealing with clones, this is overkill. Maybe your clones are disposable, just kill them, you know. But if you're dealing with a really important heirloom mother plant, I'd encourage you to put a lot of effort into making sure the thing is in fact infected and it's not something that's a fluke. Now, what was interesting about the study is we found this, and then we surveyed the roots, the leaves, the petioles, everything. The viroid is everywhere, okay? It's in the roots, and there's probably some evidence. I'm going to gamble on this and say that I think we're going to find that these things are moving through the fungal mycelium network, because there is evidence of viroids moving, hop streak virus in particular, moving in mycelium in other plants. So we have to pay attention. This might not just be about cleaning scissors. If you got plants that are in the same root beds, be careful. There may be a mechanism for these viroids to actually communicate through the fungal mycelium network. Now, we are also putting in some tools. There's a group here, Ugentech, that has some of this running outside in the exhibit hall that's going to help us with auto-calling these PCR tools, okay? When a lot of the labs that are running PathoSeq are running lots of these samples, and it's a little bit cumbersome to have people go through all of the CT scores and score them manually. So this is an AI system that was really honed over the coronavirus pandemic, where they've gone through millions and millions of samples, and they can readily call these curves automatically and flag which ones are truly failing, which ones have the internal controls functioning, that's very important, and which ones need to be rerun. So this will help automate a lot of the work that's out there. Take a peek at it over there in the booth. Now, where else can we apply this? Well, during the pandemic, I don't know if people followed this, but there's a lot of interesting work on a compound known as fluvoxamine. Fluvoxamine is an SSRI, but it's a very interesting SSRI that has sigma-1 receptor activity, somewhat unique in that regard. And if any of you follow the DMT space, you know that DMT also hits a sigma-1 receptor, and it also is a serotonin mimemic, right? So many people think psilocybin can replace some of these SSRIs. In fact, that's been published in the New England Journal of Medicine. So we got interested in sequencing this thing because it also has been shown to play some interesting roles in treatment-resistant depression and in cancer, terminal cancer depression. So a wonderful presentation today out about using this in breast cancer. So we called up Seth and said, hey, do you have any spare space in your PacBio? All of the universities in COVID seem to be shut down and they can't get us an accurate turnaround time on using the PacBio. So we sent him some high molecular weight DNA. He fitted on in one of the holes in one of his sequencers while he was doing hundreds of other cannabis genomes, and out popped this genome in remarkable shape after just being on a fraction of one of his flow cells. And it was actually remarkable. The HiFi sequencing on this had 3 of the chromosomes come out tip to tip without needing to touch them at all. That has never happened in my life in genomics. There's always been an eyeball. A human eyeball has had to take care of a lot of the repeat structures in any fungal genome. So it's now in 13 chromosomes after we put some of this Hi-C data on it from Phase Genomics. So we've got a really good annotated reference now. It's been published. It's an NCBI. It's open source for anyone to use. And we're now beginning to tease apart the completeness of the genome and some of the pathways that are in there. And the one pathway I want to turn your attention to, you'll see the Bosco scores are up there in the N50s. Those are all really important to understand the completeness. But there's, I think the best analogy here is CBG and the cannabinoids. You know, CBG is the mother cannabinoid, and then it gets split apart by other enzymes. They fight over it to decide what to make. I think there's a similar thing going on in psilocybe in that L-tryptophan is like the CBG analog, and it either makes these tryptamines that are psychoactive, like psilocybin and psilocin, originacin, baeocystin, norbaeocystin, or it makes these beta-carbolines, which are monoamine oxidase inhibitors that can impact the actual metabolism of the actual psychoactive compounds, and some of the beta-carbolines are psychoactive themselves. The beta-carbolines are predominantly made in the mycelium, and the psilocybin is predominantly made in the carpophores. So this pathway was identified by many people before us. Frick et al. And Bly et al. Had done a lot of work on this, looking at this cassette. Now, it turns out most of the genes responsible for this in Psilocybe cubensis are clustered in a 20 kb cassette that some people even cloned into brewer's yeast to try and have beer that makes psilocybin, which should be fun. But what you can do when you have sequence like this, we now have 84 of these genomes. I think there's another 16 coming off machines right now. What you can do is look at those pathway genes. This here is the PSI-M gene. This is the gene that methylates the tryptamine, or the tryptophan, okay? And what the map that you're looking at over there is a SNP EFF map that looks at how damaging the mutation is and color codes it according to how damaging it is. The really bright green ones are frame shift mutations. Those are really bad. The red ones are likely moderate mutations that you should pay attention to, and the green ones are amino acid mutations, but they're not a very high EFF score. And anything that's blue is identical to the reference sequence. So you can see in here that the reference genome that we picked, which embarrassingly is named penis envy, so if you think the cannabis names are bad, wait till you get into the mushroom space, but you'll see that it has a difference in this PSI-M gene, and maybe that's material to why it is known to make a lot more psilocybin than many of the other mushrooms. It's known to be one of the more potent ones. We have now a nice phylogenetic tree of this, and of course we can readily take a dissection into this and start looking at mating types. We know about X and Y mating types. That's very interesting. It's very similar to mammals. When you get into fungi, things get much more blurred, okay? We now have these kind of spectrums of genders that go on known as isomorphs, okay? In this case, we think this is tetrapolar and there's 4 different genders. There might be 6. We're still trying to sort that out. But this is just a picture of what some of those mating types look like. Now, what are the mating types and what govern them? They're governed by pheromones, and colonies on plates will not grow into one another if they're the wrong mating type, and they won't form clamp connections, and they won't share nuclei when this happens. And so we're now just trying to dissect these mating types. I'm a little short on time to dig into that. So we have a Cannapedia fork of this now that does the same thing. It looks at coverage maps over the key genes that are in the pathway. It tells you how related you are to everything else. But more importantly, you can dig into each variant and pull up an IGV as well. But more importantly, it also digs into the microbiome. And I want to share a story with you this last minute about why this became very important to us. The microbiome of these things is important in growing because you can't have bacteria around because it grows too fast in these things, and the fungi grow slower. So you want very sterile growth. When you have these genomes, you can build PCR assays that do look for the contamination. So this is PSI-M and PSI-K being PCR'd, looking for total aerobic count or E. coli background genomic information that may be contaminating the grow. So last few seconds, someone sent us DNA from this thing. This thing grew in 16 days. It's a monster mushroom, okay? USA Ecology had this. We went sequencing about it, and one thing that popped out are some genes that are involved in making massive mushrooms. It turns out Psilocybe, in fact, has this gene. It's on our radar, and there's very little diversity in it, with the exception of what we're seeing up there in this monster mushroom. This BM16 happens to have something weird going on inside this gene. Now, what is it? Well, we see multiple different copies of this type of gene. It's called FVCPC2. We don't yet know if this is what is driving it, but if you look through all the other versions of this gene in Psilocydia, what you find is that there are 3 dominant forms of this gene. We have 77 of the strains have one version of the gene, 5 of the ones that are outside of cubensis have a different version of the gene, and then Azurescens PS in Orissa, India have a 3rd version of this gene. And the 4th version of this gene is unique to BM16, but that we think is too good to be true. That usually tells us we could have a contamination, and it could be part of the microbiome, and that's what we have to do some work to resolve. These microbiome plots show us that when we sequence this from spores, something else was in there, and we can notice that from these blob tools plots. So we're doing more sequencing on this now to see if this is real or whether it's a contamination. Stay tuned. But the lesson of this is genomics is a very powerful tool. You can sort a lot of things out about pathogen response, about yield, and how to target the various things that may infect these organisms of interest. And I want to thank all of these people for helping us get to here. Oregon CBD is very noted up there because Seth's group's been very helpful on sequencing a lot of these things. So I'll leave it I have some time for questions. Thank you. I can't quite see out there, so I can see there's no one at the microphone. That's about all I can tell, other than maybe Ben. Okay. Do you need the stick it? Okay, so our next speaker, we are saddened to hear he's had some health issues and is staying home for this, but he did, despite all of this, put the time and effort into basically making his presentation available so that we're going to have it on recording here. This is Zamir Punja. He has done a tremendous amount of work in pathogens that infect cannabis. So it's very pertinent to what we were just discussing here. He's published many of the papers in the field. He's a real rock star in the field of the pathogens that infect cannabis. So we're going to queue up Zamir Punja's presentation next. I think we have one question before we do that.
Kevin, you mentioned with things that have the low viral count for hops latent viroid that maybe people should consider not culling and trying to keep those that might have that specifically really hard to detect low viral count for hops latent viroid. How do you recommend people safely approach that, being that the virus spreads so quickly?
Okay, so what to do about HLVD. So we did write an article about this. I'd point people to that. We think this is going to be very assay dependent, but for our assay at least, we think anything that's over a CT of 33 retest, particularly if it's a mother plant that you care about. If it's a clone, maybe it's not worth retesting because the retesting may be more expensive than just making another clone. But if it's one of your heirloom plants, everything under 33 we believe is something you probably want to pull. If you're finding something that consistently tests at 33, however, I think that's very interesting because that's a 100,000-fold lower viroid load, and what we don't want to have happen is that we, in the course of frantically screening these plants, we rip out the plants that in fact are able to manage this. It could be the fact that low viroid load is just a sign that the plant's actually clearing this and not going to be affected by it from a yield standpoint. I mean, this happens in human biology as well. When you look at coronaviruses, a lot of people get reinfected with coronavirus after they're already immune, we're vaccinated, and there's still a viroid load there, but they don't get sick. So viruses, like the presence, absence of them, I don't think is as binary as we want it to be. When your immune system is clearing these things, there will be low levels of this RNA that we can pick up for long periods of time. Even in coronavirus testing, when you're positive for 90 days, you're only infectious for 9. We don't understand that biology with this virus yet, and so I'm just putting out a little bit of caution there until we understand that biology. Retest it if it's a really important mother plant, maybe destroy it if it's in fact a clone.
So do we have a sense of whether there's an entourage effect working with psilocybin mushrooms similar to cannabis, where it's the interaction of not just psilocybin but other compounds that affects how someone feels?
Yeah. Sorry, I'm jumping down because the sound is better down there. So yes, is there an entourage effect, I think is what you're saying in psilocybin. And just like in the path of moving from olivotolic acid to CBG onto more mature cannabinoids, the same thing is going on in the psilocybin pathway where we're moving from L-tryptophan and we're decorating that molecule with different methyl groups and phosphorylating it along the way. So you get the spectrum of molecules like originacin, baeocystin, psilocin, and psilocybin. There's also norpsilocin and norbaeocystin that are different derivatives of these molecules. That's one entourage effect. The second entourage effect I think that's in the mushroom that's a little bit more interesting is that there are compounds like these beta-carbolines that we know are monoamine oxidase inhibitors that change the way that your body metabolizes the former ones, and some of those are even psychoactive themselves. So there is an entourage in these things, and we don't really know which mushrooms are making these, and our effort here is to try to tie the genotypes to those particular chemotypes over time. We want to collaborate with people that can do the chemotyping. Right now we don't know a lot of people that can do that. We're also hiring, so for folks out there, like this is part of the problem we want to solve, and we need more people for it.